Welcome back to the deep dive. Today, we're really getting into a structure that's, well, absolutely fundamental to how we move and how we're supported. Cartilage.
That's right.
We have a pretty dense stack of source material, and our goal isn't just to glance over the basics, how it cushions things. We want to achieve a real mastery of its molecular mechanics.
That really is the mission today. Cartilage often gets boiled down to just being a simple cushion, but when you look closely structurally, it's one of the most remarkable connective tissues in the body. It's this complex, incredibly durable scaffold, and here's the kicker: it's built entirely without a direct blood supply.
Which is amazing in itself.
It is. So we're going to dissect that chemical architecture, the special cells involved, and really the critical biological trade-offs that define this amazing material.
Okay, so we're going to be diving deep into the world of chondrocytes, these massive extracellular matrices, and we'll really need to differentiate between the three distinct types. That's hyaline, elastic, and fibrocartilage.
Yep, those three.
By the end of this deep dive, you should walk away knowing exactly why cartilage is so resilient, why it takes so darn long to heal if it gets damaged, and really grasp the chemistry that lets it absorb massive shocks, often for a whole lifetime.
So let's start, I think, by nailing down the core identity of this tissue. What makes it fundamentally different from, say, other connective tissues?
Right. So when we define cartilage, the key word that jumps out is durability, isn't it? We're talking about a tough, supporting connective tissue. It acts like the body's own high-performance damping material.
Precisely. Its function is just absolutely rooted in its ability to bear huge mechanical stresses.
Yeah.
Huge. And without suffering any permanent distortion.
That's critical.
Think about your spine or your knee joints, right? The sheer force transmitted through those areas day in, day out. It means the tissue has to be both elastic and resilient. And beyond just movement, it also provides that foundational support for softer tissues. It's the framework holding open your airways in the respiratory tract. And it gives shape to things like your nose and your ears.
And its role in development, that often gets overlooked, I feel. It's not just something we have as adults. It's literally the blueprint for our entire skeleton.
It really is the architectural guide. Cartilage forms that temporary fetal skeleton first,
Right.
which is then gradually replaced by bone over time through ossification. But critically, even after birth and all through adolescence, specific zones of hyaline cartilage, these things called epiphyseal plates, they guide the longitudinal growth.
So how long our bones get?
Exactly.
Well,
They determine the final length of our long bones. So it's this transitional tissue that serves vital mechanical roles both before and after the bone actually forms.
okay, so to really get a handle on its function, we have to look at the architecture. And what defines cartilage, you're saying, isn't so much the cells, but this unique extracellular matrix, the ECM.
The ECM is absolutely everything here. It's the reason cartilage has that characteristic firm, almost semi-rigid consistency. And unlike other connective tissues, it really only has one main cell type, the chondrocyte.
Just one.
Just one, yeah. And these cells sit alone in these little pockets or cavities called lacunae. Their only job really, is to synthesize and maintain all the components of this vast matrix surrounding them.
That's a huge difference from, say, dense connective tissue, where you've got fibroblasts and lots of other mobile cells wandering around.
A crucial distinction, yes.
Okay, let's talk chemistry then. This matrix isn't just like collagen fibers thrown together. It sounds more like a sophisticated hydrogel. We're looking at type 2 collagen interacting with massive amounts of, what was it, glycosaminoglycans.
Glycosaminoglycans, JAGs for short, and proteoglycans.
Yes.
And this is really where we start getting into the mastery level. The mechanical properties of cartilage, its resilience, its sort of pliancy, they don't actually come primarily from the stiffness of the collagen fibers themselves.
Oh, interesting.
Instead, they rely heavily on these other molecular players that create this incredibly hydrated environment. You have enormous concentrations of these highly negatively charged molecules, things like hyaluronan, and these JAG chains sticking out from the proteoglycan core proteins.
So wait, the type 2 collagen is kind of like the rebar, the reinforcement netting in concrete?
Good analogy.
But the JAGs are like these powerful little magnets that just grab and hold on to water.
That's a perfect analogy, yeah. These strong negative charges on the GAGs attract and bind an absolutely immense amount of water.
Right.
We're talking up to 60, maybe even 80% of the wet weight of fresh hyaline cartilage.
Wow, that much?
That much. And this bound water is what gives the matrix its turgor, its resistance to compression. When you squeeze cartilage, you're literally forcing some of that water out.
Okay.
But the moment that pressure is released, those negatively charged GAGs, they just suck the water right back in. It's this trapped, resistant water that's the real secret to its shock absorption. It creates this resilient structure that can deform under stress and then immediately spring back. And this whole system is held together by electrostatic bonds, linking the type 2 collagen fibrils to the sulfated JAGs and the long hyaluronan molecules.
We should also mention the proteins that kind of glue everything together, right? The anchors keeping the cells attached to this watery matrix they're floating in.
Yes, absolutely. Chondronectin is vital here. It's a type of structural, multi-adhesive glycoprotein.
Okay, like a specialized glue.
Think of it exactly like that, yeah. A specialized glue for this tissue. Chondronectin acts as a molecular bridge. It specifically binds to the GAGs, to the collagen, and to receptors called integrins on the surface of the chondrocytes themselves.
Connects everything.
Connects everything. This complex connection mediates how the chondrocyte adheres to its surrounding ECM, making sure the cell stays tethered and, importantly, responsive to what's happening in the matrix around it.
Okay, this brings us to what you called the great paradox of cartilage. This trade-off that gives it durability but seems to limit its biological dynamism. It's a vascular, annular, low metabolism nature.
It's the absolute defining constraint of the tissue. Cartilage universally lacks blood vessels. It lacks lymphatic vessels, and it lacks nerves.
Which seems crazy for something under so much stress.
It does seem counterintuitive, doesn't it? For a tissue handling such high mechanical loads, operating without its own dedicated circulatory system. But that seems to be the price it paid for achieving this level of structural integrity and resilience.
So, if the cells, the chondrocytes, are buried deep inside this dense matrix and there are no capillaries snaking through to deliver oxygen or nutrients, how on earth does a chondrocyte even survive?
Survival hinges entirely on diffusion. And because diffusion is slow and inefficient over long distances, chondrocytes exhibit really low metabolic activity. They have to.
They sip, they don't gulp.
Exactly. They're adapted for scarcity. Nutrients, primarily oxygen and glucose, have to slowly diffuse through that dense matrix from external sources.
And those sources are?
Typically, it's the capillaries located in the surrounding connective tissue sheath, which is called the perichondrium. Or, in the special case of articular cartilage, the cartilage in joints, the nutrients come from the synovial fluid bathing the joint space.
And this reliance on slow diffusion must mean cartilage can't just get infinitely thick, right? There's a physical limit set by how far things can diffuse.
Absolutely. That diffusion distance sets a hard physical limit. But interestingly, diffusion isn't entirely passive here. It's actually an active, movement-dependent process.
How so?
We sometimes call it the compression-decompression engine. Movement, walking, flexing your joint, to general body movement, it's vital. Because the intermittent compression of the tissue physically squeezes water and solutes out of the matrix.
Okay.
And then the subsequent decompression, when the pressure is off, allows fresh water and nutrients to flow back in. It's like a little pump system.
Like squeezing a sponge.
Precisely. If the tissue remains static, nutrient delivery basically stalls. And this biological constraint, this need for movement-driven diffusion, directly determines the maximum practical thickness you can have for functional hyaline cartilage.
Okay, let's just focus then. Let's zoom in on hyaline cartilage. This is the most common type, you said, and it's named after the Greek word for glass.
Okay.
Because it looks sort of clear.
That's right. Hylos means glass. In its fresh state, it's quite homogeneous and semi-transparent, kind of glassy looking.
And where is this sort of blueprint tissue most active in our bodies? Where do we find it?
Hyaline cartilage is really the workhorse, the mechanical foundation in many places. You find it covering the articular surfaces of basically all movable joints. That's where its incredibly smooth, low-friction surface is essential for easy movement.
Right, in the knees, hips.
Knees, hips, shoulders, elbows, exactly.
Right.
It also provides that essential, flexible skeletal support in the walls of the larger respiratory passages. Think the nose, the larynx, the trachea, the bronchi. Got to keep those airways open. We also see it as the costal cartilage, which connects the front ends of the ribs to the sternum. And as we mentioned earlier, its crucial role in the epiphyseal plates for that postnatal long bone growth. It's pretty ubiquitous.
We established that type 2 collagen is the main scaffolding, right? Making up almost 40% of the dry weight. But let's go deeper on that star molecule of the matrix you mentioned, aggrecan. What makes this particular proteoglycan such a biomechanical giant?
Aggrecan is huge, just massive on a molecular scale. It clocks in around 250 kilodaltons. But what really makes it unique are its side chains. Each single aggrecan molecule typically boasts around 150 GAG side chains, specifically chondroitin sulfate and keratin sulfate.
Okay, and those are the highly negatively charged ones.
Those are the ones, they're the key players in binding all that water we talked about.
But you said the real genius is how hundreds of these giants link together. They form some kind of superstructure. Walk us through that image.
Okay, try to visualize this. Think of a very, very long central molecule that's hyaluronan, like an incredibly long cable.
Got it.
Then, hundreds of these individual aggrecan molecules bind non-covalently all along the length of this hyaluronan cable. They're secured by special little link proteins.
Okay.
This creates a structure that looks remarkably like an enormous bottle brush. Where the hyaluronan is the wire core, the aggregans are the bristles sticking out.
And the GAGs.
And the GAGs are like the fine fibers sticking out from those bristles. So you have this immense, highly hydrated macromolecular complex. And then this whole massive structure binds further to the network of type 2 collagen fibrils running through the matrix.
Wow.
It's this highly cross-linked, integrated meshwork collagen fibrils, hyaluronan cables, hundreds of aggrecan bottle brushes that provides the tissue's tremendous strength and its unparalleled resistance to fluid loss when it gets compressed. It's an amazing piece of molecular engineering.
That level detail really explains the function. But, when you look at hyaline cartilage under a microscope, the matrix doesn't always stain evenly, does it? It suggests there's some kind of chemical difference near the cells.
That's a keen observation.
Yeah.
And it's correct. The chondrocyte is actively making and secreting this stuff, so it makes sense that the matrix immediately around it might have a slightly different composition than the matrix further away.
Right.
And we do recognize two distinct zones based on this. The territorial matrix is the area immediately surrounding each lacuna, each little cell pocket. It tends to be relatively richer in those sulfated GAGs, and a bit sparser in the structural collagen fibrils.
Okay, more GAGs near the cell.
Exactly. And because of that higher concentration of negatively charged GAGs, it often stains more basophilically, meaning it looks darker blue or purple in typical histological preparations.
And the rest of it, the bulk of the tissue between these cell territories.
That's called the interterritorial matrix. This zone is further away from the chondrocytes, and conversely, it's richer in the type 2 collagen fibrils. This forms the primary physical scaffold that separates the individual cells and their little groups.
Okay. Now, if we zoom right in on the cells themselves, the chondrocytes, we often see them in these characteristic clusters. They're called isogenous aggregates, right? Groups of maybe up to 8 cells. What's the significance of finding them clustered like that?
Those groups are basically snapshots of growth happening from within the tissue. They originate from the mitotic division of a single parent chondroblast or chondrocyte, so they're essentially clones.
Oh, okay.
They only temporarily occupy a shared lacuna right after dividing, but as they continue to actively synthesize and secrete new matrix material around themselves, they gradually push each other apart. Eventually, each daughter cell comes to occupy its own separate lacuna within the expanding matrix.
Given that they're living in this low oxygen avascular world, how do they actually fuel themselves? Do they use the typical aerobic respiration that most cells use?
They really can't rely on it heavily because they respire under such low oxygen tension. Their metabolism is specifically adapted for that scarcity. They rely predominantly on anaerobic glycolysis to metabolize glucose.
Without oxygen?
Right, without needing much oxygen. This allows them to produce the necessary ATP, the energy currency, despite the limited oxygen diffusion reaching them. And this metabolic adaptation is a key factor contributing to their relatively slow metabolic rate compared to cells bathed in blood supply.
Okay, let's properly define that perichondrium layer you mentioned earlier. It surrounds most cartilage except articular. It's more than just a wrapper, isn't it? It sounds like a life support system.
It absolutely is the life support and also the growth center for most types of cartilage. The perichondrium is essentially a sheath of dense connective tissue that forms the outer boundary, the interface between the cartilage and the tissues it's supporting.
Yeah, okay.
Critically, it harbors a vital blood supply. The capillaries and small vessels are in there, and that's what facilitates the diffusion of nutrients into the cartilage matrix to feed the chondrocytes. It also contains a small component of nerves.
And you mentioned it has layers, 2 operational layers.
Yes, typically described as having 2 layers. The outer region is primarily fibrous. It's composed mainly of dense type I collagen fibers and mature fibroblasts, giving it structural protection.
Okay.
The inner layer, sometimes called the chondrogenic layer, is more cellular and is absolutely critical for growth. It contains mesenchymal stem cells. These cells retain the potential to differentiate into new chondroblasts.
Ah, so new cartilage cells can come from there.
Exactly. These new chondroblasts then start laying down new matrix on the periphery, adding new layers of cartilage to the outside. This process is called appositional growth.
Now, here's that crucial detail again, the one that really impacts things clinically. Articular cartilage. Why is the fact that it lacks a perichondrium so significant?
It's hugely significant. The hyaline cartilage that caps the ends of bones within our movable joints, it has no perichondrium at all. Zero.
None.
None. This means two critical things. One, it has no internal source of those mesenchymal stem cells, so it can't undergo appositional growth to repair surface damage by adding layers from the outside. And two, and perhaps even more critically, it has no adjacent capillaries in a perichondrium to get nutrients from via diffusion. Its entire sustenance, oxygen, glucose, everything relies exclusively on diffusion from the synovial fluid that fills the joint space itself.
And synovial fluid isn't exactly blood.
Not at all. It's much less rich in nutrients and oxygen. This complete lack of a dedicated support system, no perichondrium, is precisely why articular cartilage is so incredibly vulnerable to wear and tear, and why it has such a limited capacity for repair, especially as we age.
Speaking of life factors, let's touch on how cartilage growth and maintenance is regulated. It seems like chondrocyte activity is under pretty tight hormonal control.
Oh, absolutely. It's under significant endocrine control. The synthesis of those key components, the sulfated GAGs, the secretion of proteoglycans like aggrecan, the very things that give cartilage its function, their production rate is accelerated or decelerated by specific hormones and growth factors.
And the major player is growth hormone, right?
Somatostatin.
Somatotropin. Yes, the growth hormone produced by the pituitary gland is a major player. And you mentioned earlier it works indirectly. How does that pathway actually function?
That's an important point. Somatotropin itself doesn't directly stimulate the chondrocytes much. Instead, it acts on the liver, primarily.
Ah, on the liver.
Yes, it promotes the liver to produce and release intermediary factors. These are known as insulin-like growth factors, or IGFs, sometimes called somatomedins.
Okay.
It's these somatomedins that then travel through the bloodstream, reach the cartilage, and directly stimulate the hyaline cartilage cells, the chondrocytes. They significantly ramp up matrix production and cell proliferation.
So it's a relay system, pituitary to liver to cartilage.
Exactly. It's an indirect signaling loop that ensures growth is coordinated across the skeletal system. And other hormones play roles too, of course, things like certain thyroid hormones, testosterone. They also modulate the overall synthesis of matrix components.
This detailed picture of matrix maintenance, how it's built up and broken down, leads us straight into the bane of aging joints, osteoarthritis. It sounds like a chronic condition rooted fundamentally in the failure of this very system we've been describing.
It is precisely that. Osteoarthritis is essentially what happens when the rate of matrix degeneration starts to outpace the body's incredibly slow, very limited repair mechanisms in articular cartilage.
It's a losing battle over time.
It often becomes one, yes. It's a chronic condition, particularly common in joints that bear a lot of weight, like knees and hips, or ones that are heavily used over many years. The initial phase usually involves the gradual loss of that articular hyaline cartilage, or significant changes in its essential biomechanical properties. The matrix might lose some of that bound water, become less resilient, stiffen up.
And then, doesn't the process sometimes turn into a kind of destructive feedback loop involving the immune system?
That's exactly what often happens, unfortunately. As the cartilage surface undergoes wear and tear, microscopic fragments of the matrix bits of type 2 collagen, pieces of the aggrecan structures get released into the synovial fluid within the joint space.
Debris.
Molecular debris, yes. And these fragments act like little distress signals. They trigger activation of nearby cells, including local immune cells like macrophages that reside in the joint lining.
Okay.
These activated cells then start secreting highly damaging enzymes, specifically a family called matrix metalloproteinases, or MMPs.
And what do MMPs do?
They chew up matrix. They actively digest and destroy the surrounding cartilage matrix components, the collagen, the proteoglycans, so they exacerbate the original damage caused by wear and tear.
So the body's attempt to clean up actually makes things worse.
In this context, yes. This destructive loop accelerates the erosion of the cartilage, leading progressively to the joint pain, the inflammation, stiffness, and eventual loss of function that are the hallmarks of osteoarthritis.
Okay, we've really established that hyaline cartilage blueprint. But you mentioned the body has adapted cartilage for different jobs, right? Sometimes you need maximum flexibility, other times you need incredible tensile strength.
Exactly. It's a beautiful example of structure perfectly matching specific functional needs. The basic cellular setup chondrocytes, chilling out in their lacunae, remains pretty much the same. But key additions or changes to the ECM composition completely alter the tissue's properties, giving us elastic cartilage and fibrocartilage.
Let's tackle elastic cartilage first. You said its composition is almost identical to hyaline, but with one really crucial molecular addition.
That's the essence of it. Think of it as hyaline cartilage plus a whole lot of elasticity. It contains the standard type 2 collagen fibrils, just like hyaline. But crucially, it also features an abundant, dense network of elastic fibers woven throughout the entire matrix.
Ah, elastic fibers, like tiny rubber bands mixed in.
Pretty much. And this addition allows the tissue to bend extensively, deform quite significantly, and then snap right back to its original shape, repeatedly, without damage. It makes it far more flexible and pliable than regular hyaline cartilage.
Which makes perfect sense for the places we find it, right?
Yeah.
Locations that need to maintain a specific shape, but also need to withstand bending or deformation constantly.
Absolutely. The body uses elastic cartilage in structures like your external ear, the auricle. Needs to be flexible, right?
Yeah.
It's also found lining the auditory tubes, the eustachian tubes, the external auditory meatus, your ear canal, the epiglottis, that flap that covers your windpipe when you swallow.
Needs to move a lot.
Needs to move a lot and spring back. And it's also in certain cartilages within the larynx. Visually, if you look at the fresh tissue, that dense network of elastic fibers often gives it a slightly yellowish color, which helps distinguish it from the more glassy, whitish-blue appearance of hyaline cartilage.
And does it have a perichondrium?
Yes. Critically, like most hyaline cartilage locations, articular being the exception, elastic cartilage always possesses a surrounding perichondrium for support and nutrients.
Okay. Now let's switch gears to fibrocartilage. This one sounds different. It's designed not so much for flexibility, but for massive resistance to tearing and high tensile stress. It sounds like a true hybrid tissue.
It really is the ultimate hybrid in the cartilage family. Fibrocartilage is best defined as an essential structural blend, a mingling of hyaline cartilage and dense connective tissue, like you find in tendons or ligaments.
Ah, okay.
Its purpose is to serve as incredibly tough, shock-absorbing, cushioning support tissue. We find it in places where movement needs to be somewhat limited, but the compressive and tensile forces are just immense. Think the spine.
So it must have a mix of cellular and molecular components that you just don't find together in hyaline or elastic cartilage. What specifically gives it that incredible tensile strength, that resistance to being pulled apart?
The matrix composition is absolutely key here. While you do still find some type 2 collagen fibers immediately surrounding the chondrocytes, just like in hyaline, the bulk of the fibrocartilage tissue contains large, highly organized bundles of type I collagen.
Type I, not type 2.
Type I. And type I collagen fibers are significantly thicker, they're more linear, and they arrange themselves into these dense, almost rope-like bundles. Think steel cables compared to the finer mesh netting of type 2 collagen.
Okay, big difference.
Huge difference. It's this bundled type I collagen, derived from dense connective tissue biology, that confers the exceptional tensile strength and the powerful resistance to shearing forces that defines fibrocartilage.
And how are the cells arranged in this very fibrous, dense environment? Are the chondrocytes still in those isogenous groups?
You do find chondrocytes, yes. They can occur singly, or often you see them arranged in these characteristic aligned sort of linear rows. We call them axial isogenous aggregates. But these pockets of chondrocytes and their surrounding sparse type 2 matrix are often separated by large regions that contain fibroblasts.
Fibroblasts. Okay, so cells from regular connective tissue are in there too.
Exactly. Fibroblasts living amongst those dense bundles of type I collagen that they themselves synthesized. So it truly is a structural and cellular hybrid of cartilage and dense connective tissue proper.
That unique composition must affect how it looks under the microscope, right? Especially concerning those proteoglycans that usually make hyaline cartilage stains so dark blue or purple?
It certainly does. Fibrocartilage typically appears distinctly more acidophilic, meaning it stains pinker or redder with common stains like H&E compared to either hyaline or elastic cartilage.
Why is that?
It's primarily due to the relative scarcity of proteoglycans in its matrix. It just doesn't have nearly as much of that GAG-rich, water-binding matrix compared to hyaline cartilage, especially compared to the territorial matrix around hyaline chondrocytes. Fibrocartilage is fundamentally a drier, much more fiber-dominated tissue.
And this super tough stuff is indispensable, isn't it? Found in the pubic symphysis, holding the pelvis together, the meniscus in the knee, that crucial C-shaped cushion. And as the primary component of our intervertebral discs, acting as those vital shock absorbers between the vertebrae and our spine.
Exactly right. And here's the other critical structural detail, similar to articular cartilage, that defines its pathology and its repair challenges. Fibrocartilage notably lacks a distinct surrounding perichondrium.
No perichondrium again.
There's no perichondrium.
Yeah.
So, like articular cartilage, it has to rely solely on diffusion from adjacent tissue boundaries for its nutrients and for any potential repair cells. And this, again, contributes significantly to its extremely slow and often very complicated repair profile when it gets injured.
Okay, we've mapped out the structure, the types, the locations. Let's turn now to how this tissue gets made in the first place, chondrogenesis, and then how it actually grows or enlarges after it's formed.
Right. So all cartilage tissue, regardless of type, originates from the same precursor tissue in the embryo, the mesenchyme.
The body's universal stem cell goo.
Kind of, yeah. The initiation of chondrogenesis, making cartilage, is actually a pretty striking example of coordinated cell behavior. First, these mesenchymal cells, which normally have these long extensions, they retract them, round up, and start multiplying really rapidly. They form these dense, tightly packed clumps or condensations of precursor cells.
So they all huddle together. That signals commitment to becoming cartilage.
That condensation is the first key step, yes. It marks the commitment to the cartilage lineage.
Huh.
Then differentiation starts happening within that clump. The cells on the periphery of the condensation become chondroblasts. They're in a phase of rapid proliferation and initial matrix secretion.
Okay.
The cells more towards the center of the clump differentiate into chondrocytes. Their primary role shifts more towards maintaining the matrix that's being laid down.
And you can tell they're busy.
Oh, yeah. Both the chondroblasts and the young chondrocytes show clear visual evidence of their intense activity. They have very basophilic cytoplasm staining dark blue, which indicates it's packed with rough endoplasmic reticulum, RER, and Golgi complexes. That's the machinery needed for synthesizing and exporting huge amounts of protein, specifically collagen and proteoglycans.
Right, the protein factories are working overtime.
Exactly. And as they continuously produce this ECM, the newly secreted matrix physically separates the chondroblasts from each other, gradually enclosing each one in its own little capsule, its lacuna.
And meanwhile, the mesenchyme around this forming cartilage chunk turns into the perichondrium.
Precisely. The superficial layer of mesenchyme just outside the main condensation gradually differentiates to form that supportive connective tissue sheath, the perichondrium, that we discussed earlier for hyaline and elastic types.
Okay, so that's how it forms initially. But once a piece of cartilage exists, the body uses two distinct mechanisms to make it bigger, right, to increase its mass or length. The first one is interstitial growth from within.
Correct. Interstitial growth relies solely on the mitotic division of pre-existing mature chondrocytes that are already embedded within the cartilage matrix.
The ones already in lacuna.
The ones already in lacuna, yes.
Yeah.
This is the process that creates those isogenous groups, the little clusters of clones we saw earlier. The cells divide, then each daughter cell starts producing new matrix around itself, and they effectively push each other apart, expanding the overall cartilage mass from the inside out.
Where is this internal growth most important?
This mechanism is absolutely crucial during development for increasing the length of long bones that happens specifically at the epiphyseal growth plates. And importantly, it's basically the only method available for trying to replace worn-out matrix in articular cartilage later in life, because articular cartilage lacks that perichondrium needed for the other growth method.
Right. And that other method, appositional growth, does rely on the presence of that outer sheath, the perichondrium.
Exactly. Appositional growth is growth from the outside in, essentially. It involves adding new layers of cartilage tissue onto the outer surface of the existing cartilage.
How does that happen?
This requires that specialized inner cellular layer of the perichondrium. Remember, it contains those mesenchymal progenitor cells. Under the right signals, these progenitor cells differentiate into new chondroblasts. These fresh chondroblasts then start secreting new matrix right there on the surface underneath the perichondrium, adding to the girth or thickness of the cartilage mass.
So appositional growth makes it thicker, interstitial growth makes it bigger from within, potentially longer.
That's a good summary. And appositional growth is generally the more important mechanism during postnatal development for increasing the overall size and shaping of most cartilage structures. But again, it's completely impossible for articular cartilage and fibrocartilage as they lack that necessary perichondrium.
Okay. This unavoidable reliance on only the slow internal interstitial growth for articular cartilage repair brings us right back to the harsh reality of cartilage injury. Why, despite being so durable initially, is its capacity for repair so notoriously, frustratingly poor?
It really is the tissue's Achilles heel, isn't it?
Yeah.
And the root cause, as we keep coming back to, lies in that fundamental biological trade-off it made.
Hmm.
Its avascularity and its intrinsically low metabolic rate.
Not enough resources, not enough speed.
Precisely. Those features fundamentally inhibit any kind of rapid biological response, including healing. When cartilage gets damaged, especially in adults, it typically undergoes incredibly slow and very often incomplete repair, if it repairs at all.
What's the typical failure mechanism we see? Like if there is a perichondrium nearby, like in hyaline cartilage that's not in a joint, what usually goes wrong when it tries to fix a tear?
Well, any significant repair often depends on those progenitor cells migrating from the perichondrium into the injured site. That's the hope. However, very frequently, those invading cells fail to successfully differentiate into functional chondrocytes capable of producing the right kind of matrix. The type 2 collagen and the JAG-rich proteoglycans needed for proper cartilage function.
So what do they make instead?
Instead, they often default to producing a scar. But it's a scar made of dense connective tissue, the kind you find in ligaments or tendons, dominated by thick bundles of type I collagen fibers.
Ah, the wrong stuff.
Exactly the wrong stuff for cartilage function. While this scar tissue might fill the physical defect and provide some structural integrity, it utterly lacks the unique elastic, compressive resistance, and shock-absorbing properties of the original type 2 hyaline cartilage. The body essentially plugs the hole, but it plugs it with the wrong material, functionally speaking.
Okay. Let's look briefly at the pathology of cartilage over a longer lifespan. It seems susceptible to calcification as we age, like turning bony.
Yes, particularly hyaline cartilage. It often succumbs to developing partial or sometimes quite isolated regions of calcification. This seems to be driven by degenerative changes occurring in the chondrocytes themselves over time.
Where does this happen most?
It's very common, particularly in areas that are perhaps less mechanically active, like the costal cartilages connecting the ribs to the sternum. Yeah. And what's fascinating from a biological perspective is that this degenerative calcification process often visually resembles endochondral ossification, which is the normal programmed process by which the fetal cartilage skeleton is systematically replaced by bone during development. So in aging, it's like an echo of that developmental process, but happening in a more patchy, degenerative way.
Interesting. And while it sounds like they're relatively uncommon, we do also see tumors that can arise from cartilage cells themselves.
Yes, that can happen. Cartilage cells, the chondrocytes or their precursors, can give rise to tumors. We generally categorize them based on their behavior. Benign, slow-growing tumors are known as chondromas. Malignant tumors, which are usually still quite slow-growing compared to many other cancers, are called chondrosarcomas.
And how are those typically handled?
Chondrosarcomas are typically managed surgically by removing the tumor. And thankfully, reflecting the generally low biological dynamism and slow turnover of the tissue they originate from, they seldom metastasize to distant sites.
Let's maybe cap off this deep dive with a couple of powerful clinical examples. Things that really show the dramatic consequences when either that type 2 collagen backbone fails or when that specialized hybrid tissue, fibrocartilage, breaks down. First, you mentioned the saddle nose deformity.
Yes, this is a really vivid, unfortunately quite graphic illustration of what happens when the type 2 collagen framework collapses. In some situations, like prolonged use of certain substances, such as cocaine, it can lead to chronic inflammation in the nasal passages, rhinitis, and critically localized ischemia, meaning poor blood flow to the tissues. This systemic damage causes the structural integrity of the nasal septum, which is primarily composed of hyaline cartilage supported by type 2 collagen, to fail catastrophically. The matrix degrades, the type 2 collagen fibers basically dissolve or break down.
The whole structure just goes.
The whole structure collapses. This leads to perforation, a hole in the nasal septum, and the visible collapse of the bridge of the nose, resulting in that characteristic saddle nose deformity. It's a stark reminder of how utterly dependent the visible form of these structures is on the microscopic molecular integrity of that type 2 scaffolding.
Wow. Okay, and secondly, let's quickly revisit the fibrocartilage and the knee, the meniscus. What happens when that super tough hybrid cushion gets damaged, maybe through injury or just long-term wear?
Right, the meniscus. Being fibrocartilage, it's specifically designed for exceptional toughness, blending both type I and type 2 collagen for both tensile strength and cushioning. However, in cases of severe trauma, like a bad sports injury or just from chronic wear associated with osteoarthritis, the original specialized tissue gets damaged.
And because it doesn't repair well?
Exactly. Because fibrocartilage also lacks a perichondrium and relies on that slow diffusion for any repair attempt, what often happens is that the original optimal blend of type 1 and type 2 collagen gets replaced over time by scar tissue.
Well,
But it's predominantly dense connective tissue scar tissue, dominated almost solely by the rapidly forming type I collagen fibers produced by fibroblasts that might invade the area.
so it loses that specialized blend.
It loses the crucial blend. Yeah. This shifts the biomechanical balance away from the ideal combination of cushioning and resilience, and more towards just rigid, less functional support. It becomes more like a ligament scar than a meniscus. And this altered tissue ultimately changes the smooth mechanics of the joint, potentially accelerating further degeneration. Hashtag tag outro.
Okay, let's try to connect all these intricate molecular dots then. We've covered well, a staggering amount of detail, really, on a tissue that most of us probably take for granted most of the time.
We really do.
From the sheer genius of using bound water trapped by that aggrecan superstructure to create an almost non-compressible biological shock absorber, all the way to the specific, deliberate inclusion of thick type I collagen bundles to achieve the incredible tensile strength needed in fibrocartilage.
I think what's most striking, when you step back, is that fundamental biological compromise we kept circling back to. Cartilage achieved its truly exceptional mechanical resilience partly by evolving to operate without blood vessels and by dialing down its metabolic rate.
Right.
But this very structure, perfectly optimized for enduring continuous load-bearing, paid a really heavy price for that durability. It sacrificed its ability to heal quickly or effectively, and that trade-off leads directly to many of the chronic degenerative conditions like osteoarthritis that plague so many people as they age.
We've really mastered those core chemical differences now, I think. Type 2 collagen forming that fine, pressure-resistant mesh that's the backbone of hyaline and elastic cartilage. But the deliberate incorporation of fibroblasts and those robust bundles of type I collagen, that's the structural key that transforms it into the super tough fibrocartilage needed in places like our intervertebral discs and meniscus.
And if we look at the bigger picture, the specific challenges faced by the chondrocytes themselves, those strict limits on nutrient diffusion, the necessary switch to mostly anaerobic glycolysis for energy, and the resulting incredibly slow biological turnover, all of these factors define the inherent limitations of this tissue type.
And that leads straight to pathology.
It leads directly to the common pathologies we see, like osteoarthritis, where the rate of matrix destruction, whether from wear or injury or inflammation, simply overwhelms the chondrocyte's sluggish, very limited repair capabilities. The balance tips towards breakdown.
So where does that leave us? We're left with this massive challenge in regenerative medicine, aren't we? A challenge that seems to rest entirely on truly mastering this tissue structure at the deepest level. Given that the body often defaults to that path of least resistance, forming a type I connective tissue scar instead of functional type 2 cartilage after an injury, what specific combination of interventions may be signaling molecules like those somatomidins? Or perhaps specially cultured mesenchymal stem cells delivered to the site? What would actually need to be introduced to truly override that natural scarring response and essentially trick the body into regenerating perfect functional hyaline cartilage?
That really is the million-dollar question, isn't it? That's the ultimate test currently facing regenerative science and tissue engineering. Can we harness the incredibly detailed molecular blueprint that we've just spent this time detailing? Can we use that knowledge to finally overcome the profound biological limitations that are inherent in cartilage's otherwise remarkably durable design? That's the mastery challenge still facing the field.
A fascinating challenge indeed. Until next time, keep diving deep into the microscopic details that define our structure and function.